Welcome to the podcast of the Journal of the American Society of Nephrology. I'm Josie Briggs, Editor-in-Chief of the Journal. Each month, we talk with authors or editors about work in the current issue. Our hope for these conversations is that they will prompt you, our listeners, to go to our website or turn to the latest issue, which should be in your mailbox very soon. This month, in the January issue, we're publishing an important study by a team of investigators in Melbourne, led by Dr. Melissa Little. The study uses kidney organoids derived from pluripotential stem cells to explore the biological mechanisms of a common genetic disease. There's a lot of excitement right now about organoid research. So I thought this work is a good opportunity to introduce to our listeners this topic and talk about why it is generating so much excitement. Dr. Melissa Little is joining me to talk about this work. Now, one quick aside, Dr. Little is both the lead of the team which performed the studies we are talking about today and an associate editor for the journal. Whenever a manuscript is submitted to Jason, where one of the authors is also a member of our editorial team, we name a guest editor, an internationally recognized expert on the topic. The guest editor selects the reviewers and makes all editorial recommendations about the work. Dr. Little was recused from editorial decisions about this specific study. However, when it comes to podcasts, I felt we couldn't avoid her incredible and relevant expertise. So welcome, Dr. Little. Dr. Little is director of the Kidney Regeneration Laboratory at Murdoch Children's Research Institute in Australia, and she's professor at the University of Copenhagen in Denmark, where she's CEO of ReNew, the Novo Nordisk Foundation Center for Studying Stem Cell Medicine. In 2022, she was president of the International Society for Stem Cell Research, and in 2021, she was a recipient of the Homer Smith Award. So extraordinary qualifications to talk about our topic today. Dr. Little, thanks for joining us.
Well, thank you, Josie. I'm honored to be here.
The paper we're going to talk about today has the following title: Kidney organoids generated using an allelic series of the NPHS2 point variants reveal distinct intracellular podocyte SYN mistrafficking. First author is Dr. Oda Dorison. So first, Dr. Little, can I ask you to start with the basics? What is the genetic disorder you and Dr. Dorison are studying here?
Well, in this study, we're really trying to look at the molecular basis of steroid-resistant nephrotic syndrome. I'm sure many of your listeners know that steroid-resistant nephrotic syndrome, congenitally inherited, is an autosomal recessive disorder, and it is actually one of the most common forms of inherited kidney disease. Children with this disorder have profound proteinuria because the podocytes efface, and this can be life-threatening. One of the genes most commonly mutated in this condition is NPHS2. The other is NPHS1 or nephrin, but NPHS2 encodes the slit diaphragm protein podocin. And it's previously been proposed that missense mutations in the gene causes the protein to mistraffic. But there are many, many different missense mutations in that gene that cause disease, and we really wanted to look at this in more detail with a more accurate model of the glomerulus, and we might come to that later. And so we actually recreated 5 distinct clinically associated missense mutations using CRISPR-Cas9 gene editing on the same parental background line, so the same genetic background, and then modeled the disease for each of these mutants using kidney organoids.
So the basic question here is, how do these different missense variants actually cause disease? Now, give our listeners help with a second basic question. What actually is a kidney organoid?
Yeah, it's an interesting term. So a kidney organoid is actually a multicellular microtissue model of the human kidney, and we can generate these from human pluripotent stem cell lines, whether that's ES cell lines, embryonic stem cell lines, or induced pluripotent stem cell lines. And the protocol of generating a tissue from a pluripotent stem cell line is to direct their differentiation through the steps that they would normally take to form that tissue. So in the case of kidney, we direct the cells through mesodermal specification to the intermediate mesoderm, and then they start to form a kidney. And what you end up with is actually a very accurate 3D multicellular tissue that has patterning and segmenting nephrons that include a glomeruli, plus the surrounding stroma, endothelial cells, and most of the components you would expect in a kidney within a 3-week period in the laboratory. I think what's really important for this project is that the kidney organoids contain very beautiful forming glomeruli. Now, they're immature, but they are 3-dimensional. They have a bone space surrounded with parietal epithelium. They've got podocytes. The podocytes are differentiating, polarizing, and starting to form foot processes in 3 dimensions. And this actually makes them a really ideal model to look at podocyte disease. In fact, a previous postdoc in my laboratory, Lorna Hale, spent a lot of time looking at and comparing the podocytes from kidney organoids with the immortalized podocytes cultured in 2 dimensions. And these are a much more accurate model of the podocyte.
Right. I wish we could show our listeners pictures. The pictures tell part of the story. So, of course, there's transcriptomics. These are amazing glomeruli to grow out of pluripotent stem cells. So clearly this organoid tool is being seen in many areas as a really breakthrough method to get at questions. A lot of the first applications were the relation with developmental biology, but I think we heard from the reviewers of this that this is a really important and new way to understand genetic disorders. Could you help our listeners also explain why is this a better way? What are the aspects of this that make this a more powerful way than the more conventional tools to study these genetic missense mutations in critical proteins in the podocyte?
You know, I think the podocyte field, I think nephrologists have realized for a long time the podocyte is a very complex cell type. It's very 3-dimensionally complex. And I think there's quite a lot appreciated that there's quite a lot of difficulty culturing podocytes in vitro. And while immortalization was a great advance to enable podocyte culture in 2 dimensions, they really don't express all of the podocyte markers at the level a normal podocyte does. They don't interact with each other in the way they would in 3 dimensions. And so I think examining a mutant gene in those models has been quite difficult and sometimes inaccurate. So there have been original studies looking at podocin mistrafficking in the presence of mutations, but they've always had to overexpress the gene off a non-endogenous promoter, which often leads to very high levels of the mutant protein. And that can give you incorrect information. But in our approach, because we can actually edit the stem cell and then make the stem cell into a 3-dimensional glomerulus where the podocytes are much more accurately patterned, we can create the point mutation in the genes themselves. So we actually put the missense mutation into both alleles of the endogenous gene. Then we can really look at what would happen in a patient because there's no exogenous protein added. We're really just looking at a model of the patient. And we're also able to therefore put each of these point mutations in homozygous state on the same genetic background. That actually cleans up the issue of genetic background. And that gives us a much cleaner biological model of the disease.
Right. No, it's a very exciting approach, and the way in which the CRISPR-Cas methods are contributing to this is also in itself a very exciting story. So what were the surprises here? What were the key findings?
Well, I think our first finding was that because we were hitting the gene itself and not overexpressing a mutant gene, our first finding was that all of the missense mutations, no matter which they were expressed at much lower levels at the protein level. The protein levels were depleted for all of these mutants, and that actually, and that is not at the gene level. The gene expression was normal, but the protein was down, and that suggests that a single base change in that protein gives you some problem either in the protein translation or an increase in the degradation of the protein. So that's very important, I think, and we would not have revealed that necessarily without doing it this way. We also confirmed what had been suggested, and that is that the subcellular location of each missense protein was inappropriate. So the amount of podophin reaching the plasma membrane out where it should be was reduced, but each point mutation was in a different place. So indeed, some of them were accumulating in the endoplasmic reticulum, but we had others that were specifically stuck in the Golgi complex or in endosomal compartments. And so some of them showed a small amount of trafficking to the plasma membrane, but significant mislocalization. And we were able to actually accurately quantify that. And I think that really gives us a sense that they're all acting differently, or the problem is different depending upon point mutant, which is important if you start to look for treatments. We did a lot of expression profiling. This is a standard approach. Let's have a look at the transcriptional profile and see how it's changed. And we did this very stringently across all mutants, including a patient line and a control, and there was nothing that associated with disease. And so what we're really seeing is something that's impacting protein translational stability, not impacting gene expression. And even in the lines where there was ER stress, even in the lines where there was ER mislocalization, there was no evidence of ER stress. And I think the other thing that was really nice is we could look at where nephrin was, because nephrin and podocin bind to each other at the plasma membrane, and there's been a lot of suggestion that the two of them move through the trafficking machinery together. But actually, we saw some cases where the nephrin was staying with the podocin and was stuck, and some cases where there was nephrin out of the membrane and podocin in the trafficking machinery. So I think now we've got a really wide variety of effects from different single point mutants in the same gene.
How might this, so given a patient with a specific mutation, how do you imagine ultimately one would hope to use this? What would the practical application be?
Yeah, so we've shown a number of disease states for genetic kidney disease, for inherited kidney disease, that kidney organoids can model the condition. But I think what I really would like to see is that organoid biology is used to find new treatments. Now, not all of these steroid-resistant nephrotic syndrome patients have missense mutations in MPHS2. In fact, some of them are nonsense mutations. But we chose the missense mutations for a reason, because we know in other diseases like cystic fibrosis that if you know what's going wrong with a missense mutation in terms of trafficking, you have the chance to find drugs that can assist the protein to actually get to the right place, for example, chaperones, or make it more stable or assist its functionality. And so now we know what each mutant is doing and where they're stuck. We hope to start screening drugs to see if we can find compounds that will assist in either increasing the amount of podocin protein present and or trafficking it to the right place. And so this will be what we try and do next. We are moving to trying to make even smaller and smaller versions of our glomerulus model so that we can try and do drug screening. And the hope is that we'll be able to find a therapy for congenital nephrotic syndrome.
Right. And the therapy might, in fact, have to be selected for the particular missense mutation.
That's right. Yeah, we would predict that a little bit like cystic fibrosis, that the drug may have to match the point mutation.
Right. Well, this is really important and fascinating work. I want to congratulate you on this really extraordinary study. I'm really enjoying having you as part of our editorial team as when we look at developmental biology and regenerative medicine applications, including organoid work. I think we are publishing some very strong work in this field. We also have had the benefit of input from 2 editorial fellows who've worked with you first, former trainee Tom Forbes, our current other developmental biologist on the team is Niels Lundstrom, also an Australian, and I think that the extraordinary young scientists are really helping our whole work. It's a bit of a golden age for the study of the development of the biology. I worked on this a long time ago. I was curious what the lineage was of the macula densa cell. But we're really seeing work that's at a new level of mechanistic understanding. And equally important is growing recognition that this work is getting closer to having practical application to both study of genetic disease and maybe at some point to kidney regeneration. Give our listeners a few thoughts to take away on the dreams of the next steps.
Yeah, thanks, Josie. I would just correct you. Niels Lindstrom is a Dane, and he'll probably be very keen to have that corrected.
Okay, sorry. I thought he was at least somebody. I am correct. I am corrected. I thought at least sometimes he was in Australia.
No.
Thank you for the correction. That's fine.
Tom's an Australian.
But he's
and I just...
Okay.
Sorry.
No, go ahead.
I just acknowledge Ord Dorison before I wrap up, because Ord really is a postdoc in my group that drove this study, and in fact we had to resubmit while Ord was on maternity leave having her little boy Theo, and she'll probably be listening. She's in Europe at the moment on leave, so Ord did a fabulous job. So in terms of what I think is going to happen into the future, so I think that for modelling inherited kidney disease, the organoids are going to be powerful. I think we have to be a little cautious overstepping the mark and imagining that these can be models of late onset adult chronic kidney disease, because this is a developmental model. We're very keen on using kidney organoids for drug screening, and I think that now we're starting to move towards that in a number of labs. Not just for congenital nephrotic syndrome, but for ARPKD and for ciliopathies. There's so many genetic kidney diseases that could be looked at. I think on the kind of bigger, audacious goals of can we ever use pluripotent stem cells to make functional renal tissue, this is such, from a developmental perspective, this is such a big ask, and there remain lots of challenges. But I'm up for the challenge, and we've really done a lot of work on scaling out and scaling up options, because of course we've got to get very large numbers of nephrons if it's ever going to be of any use to patients. And now we're focusing on functionalisation after transplantation. So it is a very big ask, but we and others are giving it a go, and I guess I didn't imagine we would ever be able to make an accurate three-dimensional multicellular model of the human kidney, and we have. So I think I'm very excited about where the future might take us.
Well, I am too, and I've really enjoyed having your input to Jason as we look at this kind of work. So thank you again for joining this conversation, and thank you for the fabulous work you're doing.
Thanks, Josie.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition, or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.